Thanks everyone for joining our cell therapy and solid tumors panel today. I'm very happy to be hosting what is a very complex subject, as I'm learning, and also clearly the holy grail, if it were in cell therapy. Just as an introduction, my name is Amanda Murphy. I am a biotech analyst at BTIG. BTIG is a global provider of liquidity as well as a full-service investment bank co-headquartered in San Fran and New York. We have 4 biotech analysts, of which I am one, and we also have Tom Schrader, Bert Hazlett, and Tim Chang, who are wandering around here, so say hello as you see them. I'm going to turn it over to the panel to do a quick introduction, and then we'll start with our topics.
Oh. I think I'm mic'd up.
Mic, so
yep. Good afternoon, everybody. It's always a challenge to be the first session after lunch, so get your coffee, please. My name is Thorsten Melcher. I am with a company called Xyphos. We are a startup company in the San Francisco Bay Area, 7 people. We develop a novel technology for CAR-Ts that allows us to very tightly regulate the activity of our cells, and that has the ability of built-in combination therapy treatment so that we could provide cytokine stimulation or checkpoint blockade to our CAR-T cells. We'll start out in the hematologic disease space, but then move very quickly, hopefully, into the solid tumor space. Thanks.
Good afternoon. I'm Stewart Abbot from Adicet Bio. I'm the Chief Operating Officer with the company. Adicet was established back in late 2015, and we are predominantly a gamma delta T cell company. We're a CAR T cell company focusing on gamma delta T cells. We also have capability in target identification and also in targeting moiety development, specifically antibody development. Company is based in Menlo Park. We have an affiliate office in Haifa in Israel, and we're currently about 65 people. And we'll tell you a little bit more about the approach as we go through some of the questions today.
Okay, thank you for the opportunity to be here. My name's Gunter. I'm the Chief Medical Officer at Medica. Medica is a company based in London and Houston. We are a cellular therapy company, as you might have guessed by our name. We're focusing on NKT CARs. So NKT cells are a type of T cell that is uniquely suited for treatment of solid tumor and also uniquely suited for allogeneic therapy. So it's a pleasure to be here and share some of those thoughts. We are working closely with Baylor College of Medicine and also Cell and Gene Therapy Catapult in the UK.
Great, and good afternoon, everyone. My name is Christopher Haqq, and I'm the Chief Scientific Officer for Atara Biotherapeutics. We're a South San Francisco and Thousand Oaks, California-based company, now of almost 300 staff. And our lead platform is a viral-directed T cell that we're now interested to leverage as a novel off-the-shelf platform for CAR-T therapies, directed both at solid tumors and at hematologic malignancies. We've brought in a whole toolkit of technologies that will allow us to not only make off-the-shelf CARs, but actually make a sort of a next-generation approach for these therapies in partnerships with Sloan Kettering, where we work with Prasad Adusamili and Michelle Sadelain, as well as at the Moffitt Cancer Center in Tampa, Florida, where we work with Marco Davila. It's a pleasure to be here.
Thanks, and we're very lucky to have a wide variety of approaches represented on the panel. I thought, though, it might be helpful to start with just a couple of high-level, sort of more industry questions. One, of course, being, obviously cell therapy and heme has been, the responses have been quite amazing. Maybe not the case as much in solid tumor. So just thought it might be good to go through a little bit of a history lesson in terms of, why hasn't solid tumor worked as well. And then, hopefully as you can see in the agenda, we'll kind of work our way into the specific hurdles and eventually, each of your strategies. But maybe starting out with just your perspectives on why it hasn't been as successful as heme.
Lead on. either off? Okay, I'll kick it off. There we go. History of cell therapy in solid tumors. Well, I guess history tends to be written by the victor. We haven't actually had a victor yet with solid tumors. We've won some small battles, I think, along the way, but there's quite some considerable challenges. Obviously, certainly cell therapy, CAR T cell therapy has kicked off in hematological malignancies. Lots of different reasons for that. Some great targets there, possibility of eradicating tumor without having to worry about too much about the effects on normal cell types. Obviously, you do eradicate normal cell types when targeting B cells or when generally targeting B cells, but it's not such a large issue as potentially hitting normal tissue when targeting solid tumors. There are a lot of features of hematological malignancies that actually make them quite tractable to CAR Ts. They present a lot of co-stimulatory molecules that are helpful in stimulating the CAR T cell population. Not all solid tumors do that. And they're generally pretty accessible. A lot of the liquid tumor pool is actually within the circulation. You don't have to get your CAR T cell to get into a specific specialized tissue in order to have a quite potent effect. So I think there's a lot of attributes of liquid tumors, hematological tumors, that have been made them quite tractable in the first instance. But we've learned a lot of lessons along the way that I think a lot of us are now applying to solid tumors.
I guess I'm the next victim. So the way I think about it is, there have been some hints that immune cellular therapies can be active against solid tumors, and maybe the first hint came from Steve Rosenberg's lab. That wasn't a CAR cell, but those were TIL cells, and clearly in melanoma and possibly in other tumors, those cells are quite active. Now, you could argue melanoma is a unique tumor and very susceptible to immune perturbation. Nevertheless, with other targets like HER2, with GD2, there's been some success. But I think we'd all agree in this room that the success with hematological tumors has been greater. Why is that? Well, one simple way to think about it is that you're using a therapeutic that naturally lives in the hemolymphatic system. Lymphocytes would be, you would think, more apt to traffic within the lymphoid system, and therefore lymphomas and leukemias through the bone marrow would be more susceptible to treatment. So with solid tumors, probably some of the barriers that we have to overcome include, first of all, making sure that the immune cellular therapy, whatever it may be, actually sees the chemokines that are being secreted by the tumor. And often, probably there's a mismatch between chemokines secreted by the tumor and the receptors on the cells. So they have to be attracted to the tumor. A second point is that the cells, the immune cellular therapy, has to be able to confront and overcome the barrier to actually enter the tumor. And there are several different barriers, but that's an important issue. And then finally, once the immune cellular therapy gets into the tumor, that has to overcome the multitude of forces that really suppress the immune system within the tumor. And these things range from specific ligand interactions to non-specific things like simple hypoxia. So all those factors are really unique, in my view, to solid tumors and not faced by the cell therapists that are working with hematological tumors. That sounds somewhat pessimistic, but I am optimistic that there are going to be solutions to solving many of these problems. So I'll stop now.
I'll just move forward quickly, just because you brought up some of these points in terms of some of the major issues. Obviously, target identification being one of them. Chris, you talked about having a viral, target methodology. Maybe you could start off with just how you think about building a pipeline. Obviously, given the challenges of finding targets that are, differentially expressed, et cetera.
Sure. I think Amanda's really bringing up one of the most fundamental differences between hematologic tumors and solid tumors, which is the availability of a target antigen that can be safely eliminated. And we know that many solid tumor targets are ones where there's a higher level of expression on the solid cancer, but nonetheless a lower level that's more modest of expression on normal tissues that one wouldn't want to affect because that would cause side effects in patients. So in order to target solid tumors through antigens that have that feature, one has to very carefully develop the targeting antibody for a CAR approach so that the binding reagent doesn't interact with low density expression on a normal cell, but is completely specific for the higher levels of expression that occur on solid tumors. And there's an example of that in some of the work that we do at Atara Bio in targeting mesothelin, where that property of the antibody is essential for the approach. And in addition, I'm also very enthusiastic about the prospects for the adoptive cell therapy in solid tumors because of observations that have been made with viral cells. So I like to think of this as, sort of putting together a Reese's peanut butter cup and putting chocolate together with peanut butter. And the reason I say that is because even in viral targeted T cells, we've seen in some solid cancers like nasopharyngeal cancer, which are driven by Epstein-Barr virus, that the adoptively transferred T cells, the same ones that we would use with great effect in post-transplant lymphomas, they're active, and we've seen complete response as well as partial responses occur in patients with just monotherapy with EBV-specific cells. And those cells are now in a trial where we're combining with Merck support with pembrolizumab. And that combination of checkpoint inhibition is also one of the key features that could be a theme to the slide that's currently up, where we see more exhaustion of adoptively transferred T cells that enter into that solid tumor microenvironment where they're given by themselves. And I think over the coming years, you're going to see a lot of innovation in this arena that will endow the T cells with that ability to sort of act like a Trojan horse and bring into the tumor microenvironment some other feature, molecular feature, that can help to deal with those immunosuppressive features within the tumor environment.
So I wanted to give everyone a chance on the panel to sort of talk about your specific strategies. Obviously, lots of different cell types being discussed and pursued, as well as different even CAR T strategies. So considering all the conversations we've had around target identification, tumor microenvironment, trafficking, immunosuppression, persistence, maybe we could just go through each one of you and give you a chance to discuss your company's plan.
Okay, so I can kick it off. So we are building a technology platform that is trying to address essentially all of the challenges in solid tumors that were mentioned already. Doesn't mean that a product that would be derived from that platform would have all of these elements in there. And as I said, we are still preclinical, so this is very early in the overall process. But the way our system works is that there is a universal chimeric antigen receptor that we engineered so that it does not bind any other protein. And then we also engineered the bioorthogonal ligand to it that binds with very high affinity and specificity to that receptor. And so what that receptor essentially is on the surface, on the CAR T cell, is like a mailing address where you can send packages to. And those packages could be a tumor-targeting antibody or two tumor-targeting antibodies, or it could be molecules that have the ability to modulate the function of the T cell. Like, for example, we can very specifically administer a mutated interleukin-2, and it would only act on our CAR T cells. So when we go through and I'm always thinking about the 3 T's that were mentioned, so that is the trafficking, the targeting, and the tumor microenvironment. With that kind of system, we have principally the ability to put combinations together to address all of those different factors. For example, antigen escape. If we took a B-cell malignancy, we target initially CD20, the tumor recurs, but is CD20 negative. With our system, we would dose now a CD20 targeted antibody but we don't need to manufacture another CAR T cell. So the whole philosophy is to allow the physician to control a population of CAR T cells that are in the patient from the outside by administering bispecific molecules. And these bispecific molecules being able to target a different antigen or to target two different antigens at the same time, or to somehow functionally modulate the ability of the CAR T cell. For example, around tumor microenvironment, we are having an anti-PD-1 molecule that specifically and exclusively acts on our CAR T cells. Or we are working on finding molecules that could deplete immunosuppressive cells in the tumor microenvironment. And so again, the philosophy being build a toolkit of all of those different elements, and then most likely through early clinical studies that are hypothesis-driven, put those components together that are needed for a product that is successful in the solid tumor space.
So just to give a little bit of background on why Adicet's doing what Adicet's doing, I was particularly intrigued back in 2010 by what was becoming the first of the designer cell therapies, or a designed cell therapy. And these were the CTL19s that were being developed out of City of Hope, Memorial, Sloan Kettering, NCI, and the like. Now, these had been in development for several decades before, going back to Zaligesho's work and others, but they were one of the first examples where the therapy started with the cell therapy started with a really solid problem statement. Problem statement being, have to get rid of malignant B cells. How do you get rid of malignant B cells? Well, you have to be able to target them effectively. What can target them effectively and destroy those malignant B cells? Well, T cells can, as long as they actually have a method of recognizing those cells. And that's really where what was originally T-bodies and now chimeric antigen receptors came from. They were targeting systems that allowed this target to be added to a payload, which happened to be a T lymphocyte, to go around the body and blow up B cells. If we start now thinking about the problem statement for solid tumors, as quite rightly highlighted earlier, we have to be able to get cells into the tumor. We have to be able to evade the microenvironment. We have to be able to find targets that are specific to the solid tumors and not expressed on normal tissues. We have to be able to overcome potential for antigen escape. That's quite a list of problems, and so when we thought about those problems, we realized there's only so many of those problems you can solve through genetic engineering. And so one of the things that one ought to do is try and find the very best cell for the job that can overcome certain of those problems, and then use your genetic engineering budget to solve the rest of the problems. And so that's what we did. We looked around at different cell populations, whether they be natural killer cells, NKs, NKTs, that Kurt will tell us about, alpha beta T cells, which are the majority of the cells that are out there, or most of the cells in development, and gamma delta T cells was this little underexplored, underappreciated cell population, makes up about 1 to 5% of the circulating T cells. It's a relatively low frequency, happens to be present in tissues at very high frequency, but it circulates in low frequency. But it also correlates very, very strongly, or the presence of gamma delta T cells correlate incredibly strongly with good clinical outcomes in a number of different disorders. And so we were intrigued by the potential of this gamma delta T cell to be a better T cell as a foundation for genetic engineering to then overcome some of these other challenges. The other thing that we were intrigued about was the fact that alpha beta T cells, when transferred between unrelated donors, would ordinarily create something called graft versus host disease, or GVHD, which is a huge problem for hematopoietic stem cell transplantation, and is also a problem for adoptive transfer of T cell therapeutics, or alpha beta T cell therapeutics, without considerable levels of gene editing. Turns out that NK cells, gamma delta T cells, I presume NKT cells as well, can actually be transferred from donor to donor without any tissue type matching, and they do not induce graft versus host. So you don't have to spend any of your genetic engineering budget to overcome graft versus host disease, which was key to us. And so we now have this cell type that hangs out in tissues that can get into tumors, doesn't cause graft versus host disease, happens to be highly potent when you can get them in, make sufficient numbers of these cells at clearing tumors. And so we set out to find ways of making these cells. Historically, people have been able to make gamma delta T cells, but they've only been able to make one specific subpopulation called gamma delta 2. And the reason that could be achieved was that it was realized several years ago that bisphosphonate drugs happen to drive the expansion of gamma delta T cells. We didn't want to use bisphosphonates, but we took some lessons from the alpha beta T cell world where we used specific antibodies to expand gamma delta 1 cells, another subpopulation that we find is the most potent subpopulation, or one of the more potent subpopulations. And so we generated a series of antibodies that selectively expand gamma delta T cells, maintain their potency, they don't get exhausted, they don't induce GVHD, and then only thereafter do we engineer into the cells a targeting capacity. And one of our first candidates is targeting CD20. We happen to think it's a really interesting target. It's also a de-risked target. We have a first-in-human study starting next year. It has a novel cell, novel engineering. We wanted to use a fairly well-understood target. But beyond that, as I mentioned earlier, we were looking at how do we selectively target solid tumors. And we took lessons from TCR, or T-cell receptors, in that T-cell receptors can recognize solid tumors by virtue of recognizing peptides, solid tumor peptides that are presented by HLA, or human leukocyte antigen class 1. For a lot of different reasons, we didn't want to develop TCRs, other groups have, but we wanted to develop antibodies that behave a lot like TCRs, and we call them TCR-like antibodies. They specifically recognize solid tumor-derived peptides that are presented by HLA-A2 on the surface of the solid tumor. And a lot of screening to identify those particular targets that are unique to solid tumors and not expressed on normal tissue. And that, in combination with the gamma delta T cells, allows us to go after a whole range of different solid tumor targets. So we think that the challenges can be overcome. We, like many others, have cured cancer in a mouse. That's not a high hurdle. But we hopefully will get closer to the relevant species this time next year.
Okay, well, I guess that's a hard act to follow.
You've got a good cell type.
Yeah, we do. I think ours is better than yours, too. Actually, having said that, I certainly wish everyone up here well, because we're kind of all in this industry together. And I would like, all the companies to be successful. But nevertheless, you do have to stake out your own turf. And we looked at some of the same issues Stewart was just talking about, and we came up with NKT Cells as a good biological platform. And as a small company, you just can't cover all bases, so you have to take your best shot and go with it. And what we like about NKT Cells is we can take advantage of some fundamental biological properties. Now, NKT cells are rare in the peripheral blood. In fact, they're probably even more rare than a gamma delta cell. So that's an issue. But Leonid Metalits's lab, I know that's a mouthful. I can give you the paper later if you want. But he's a guy at Baylor, a very preeminent scientist, knows NKT cells. We partnered with them, and he figured out how to grow these cells and expand them in very large numbers. And I think that was what the final step in convincing us to move forward with this. So manufacturing is a key issue for us. Now, what's unique about NKT cells? Well, first of all, they do tend to be tissue homing cells. So we think they'll probably also be tumor homing cells. So they're relatively more abundant in the tissues than the blood. And relatively is an important point because they're still rather rare. We also know that they have an invariant T-cell receptor. So Stewart's already explained what graft versus host disease is. By an invariant T-cell receptor, I mean that all the T-cell receptors and all the NKT cells in my body and in all of you are exactly the same. So if you took my NKT cells out and gave them to you, you will not get graft versus host disease. And hopefully your tumor will be cured too. So that's a fundamental advantage of an NKT cell. Unlike an alpha beta, and I say beta, not beta, excuse me, but unlike an alpha beta T cell, with a gamma delta or with an NKT cell, you don't have to edit the T cell receptor And that's a step you can avoid in the manufacturing process and potentially a safety issue that you don't have to deal with. There are some other aspects of NKT cells that make them appealing. They will be active in hematological tumors as well because they recognize a molecule through their native TCR that's expressed in hematological tumors called CD1D. So for all those reasons, we wanted to go after this as our cell platform. And we do have, through our manufacturing partnership with Baylor, access to a number of different targets. GD2 for neuroblastoma, and we're in a clinical trial with that now. CD19, we're starting a clinical trial later this year, and that. CSPG4, which is expressed in a number of tumors. GPC3, which is expressed on hepatocellular carcinoma. And finally, BCMA, which is well known to, I assume, most of you, and validated for CAR T-cell therapy now. We also, through our manufacturing relationship with Cell and Gene Therapy Catapult in the UK, have our own people in their manufacturing facility in Stevenage, and that's where we intend to make products for post-phase 1 development. So that's our plan. Chris?
Great. Well, I think you've already heard one of the key things that all of these take as a theme is a way to eliminate graft versus host disease. And so the reason that we at Atara like the viral cells is that their receptors then function in recognition of a virus. And you can eliminate all the EBV in your body without any safety concern. We're also leveraging now almost 200 patients' experience in providing this therapy for the post-transplant lymphomas to patients in numerous countries. And since our programs are in phase 3 for that platform technology, we've already established with EMA, FDA, global regulators what the criteria should be to provide that product around the world. Like you've heard, one of the key things in being able to move these rapidly forward is to have access to the manufacturing technology. And so we've invested in constructing a $60 million plant, over 100,000 square foot floor area in California, so that we can really rapidly deploy both our existing product candidates as well as the new CAR Ts that come in. And I will just say a couple of words about the EBV cells. So they're an alpha beta kind of a T cell. But what's very unique about them is that unlike the T cells that recognize endogenous genes, like those that Dr. Rosenberg's lab at NIH have pioneered in melanoma that was mentioned earlier, those have to be given on the order of 1 billion cells for each patient. And orders of magnitude lower numbers of the viral cells are required because over the course of evolution, our immune systems have evolved to recognize that those viral responses are very special. They shouldn't be interrupted. In laboratory tests, they're not susceptible to T regulatory cells or myeloid-derived suppressor cells. And they also have a really nice trafficking feature in that Rosado and colleagues have just published in one of the Nature journals this past month that viral cells, including the EBV cells, are able to traffic in the same proportion into solid tumors. They're found in any type of solid tumor present, and in her paper she showed that she could activate those and actually eliminate the tumors in a mouse, right? So we really consider that that's an ideal platform to then introduce the CARs into for solid tumors, because they already have access in trafficking, and they have a number of these special properties and can be given at low dose. We can manufacture relatively smaller amounts of them to be able to provide therapy.
Thanks. And obviously we've already started to get into the manufacturing discussion, but I did want to also talk about, allo versus auto. It seems to be a nice, healthy debate even within the investor community. I think many of you are sort of ultimately pursuing an allo approach. So I'm not sure who's willing to jump in on a broader discussion on how this might evolve. Do we see sort of a fully allo world, or is there different? Might we see a mix of approaches based on indication? How do you see this playing out? And then, if you also want to comment on your own, company strategies there as well.
Yeah. So talking for us, and I think I mentioned we are 7 people. So focus is something that is incredibly important, and we decided to really focus on our core technology, which is how to control the activity of a CAR T cell through our universal chimeric antigen receptor and this family of bispecific kind of steering molecules that I described. And we made the decision that everything else outside of that, we will try to use kind of established methodology and technology as much as that's possible. So we are going autologous into the clinic, and we are not innovating on the manufacturing side. We are not innovating on the cell side. However, of course, we are very aware that if our control and combination therapy technology works, it would be highly synergistic to a more allogeneic cell, because then you would really arrive at the status of one cell, all targets, all patients, all functions. And so our strategy is we got to validate our technology in the clinic, we got to demonstrate the potential, and we would then expect that people with more advanced cell therapy technologies, like my colleagues here on the panel, would be interested in partnering in some fashion with us.
So I think there are a couple of different thought processes that go on around autologous or patient-specific therapies versus allogeneic therapies. One of the early discussions that went on in this field was basically the cost of goods. Patient-specific therapies, generally autologous, fairly high cost of goods, therefore had to have fairly high reimbursements to make this justifiable. Expanded allogeneic, and what I mean by that is allogeneic products where you're taking cells from a healthy donor or a donor, expanding them massively during manufacture, and then giving that same batch of drug product to many, many patients are certainly more cost effective. But that wasn't actually the primary driver for Adicet developing a healthy donor-derived expanded T-cell therapy. The key drivers for us were that patient-specific therapies suffer from one really critical problem. Many patient-derived lymphocytes are dysfunctional. So a lot of patients come to CAR T-cell therapy after many years of traditional therapy. Many of those traditional therapies, including chemotherapeutics, causes a global immune system dysfunction. That can make manufacturing products from the patient's own material quite challenging. And even when you can manufacture those products, it's actually quite difficult to get consistency from patient to patient, because you're starting with starting materials that are fundamentally different from each individual patient. And so not here, but in other sessions today, we've talked about therapeutic index or therapeutic window, that difference between the effective dose of a cell therapy and the toxic dose of a cell therapy. When you have differences in your starting material, it's very, very difficult to predict what that window is going to look like. Where you have consistency in your starting material from healthy donors, it's easier. It's not straightforward, but it certainly is easier. And so we're developing healthy donor-derived expanded products, gamma delta products, primarily for consistency, but also to overcome the issues of immune dysfunction that exist in patient-specific products.
So, Stewart's so well-spoken, it's difficult for me to follow up, and he covered some of the things that I wanted to cover, but I'll try to say something intelligent here. So, I agree with the points that were made about the effects of pretreatment on patients and how that impacts the quality of autologous products. I think that's a very important issue. Also, the complexity and the costs of dealing with autologous products is also an important issue. So our bias at Cell Medica is that allo is the way to go. Now, so if we have let's just say hypothetically we had two different products, and if we got it to work in the allo setting, we'd obviously want to develop that one, because we think the economics and the quality issues will far outweigh what we could get with an autologous product. So we're strongly biased toward allo. I don't know if allo will be successful. Certainly autologous is already on the market, and those products are extremely active. And so allo is behind in the race. Now, when you're making products from healthy donors, you are probably going to have a more consistent product, but you're still going to have to go back to donors every now and then. We can make hundreds of doses from one donor, but that's probably not going to be enough to satisfy the market demand. So you're still going to have procedures in place and release criteria that define your product so you can make a consistent product from allo donor A versus allo donor B. I mean, the ultimate solution to this might be to use an iPS cell and a cell bank, but that could be many, many years away. And so for us, we think the short-term solution is relying on healthy donors. And I think that's all I want to add at this point, considering we have only 9 minutes left, and I want to give time for my colleague Chris here.
Sure. So I'll just say very briefly, because I think a lot of the points have already been covered, but we really face an extreme case of this problem of not having great manufacturing cells in the post-transplant lymphoma patients. So they've just been through the massive chemotherapy conditioning regimen for a bone marrow transplant, which actually wipes out all the cells. So you couldn't manufacture an autologous product for them. Everything that we do there is allogeneic. And what's more is that survival after rituximab failure is quite short. So on the order in different series, 2 to 5 weeks. So they wouldn't even have time to wait around for autologous manufacturing. We can give our cells within 3 to 5 days. We have a system, a web-based system where the physicians can input the HLA type. We do match the tissue type of the cells that we contain in a library in order to do that. But that's sort of well worked out. And I think I'll probably just leave it there for right now. I think it is fine from my perspective to take a lot of the steps in solid tumors first from an autologous CAR point of view. I think that's very useful to learn. And then, as my colleagues have said, if you can get it to work in an allo format, then that's ultimately what's going to win, because that will allow you to provide therapy for so many patients. And as we think about solid tumors that are very common, like colon cancers, lung cancers, ovarian cancers, things like that, we'll need to have that scalability in order to provide therapy to those people in need.
I just wanted to add one thing about Aloe Products. I forgot to mention this, but even if you're using a fully humanized construct on your immune cell therapy, an Aloe product could potentially be immunogenic because it's an Aloe cell. And so that's something that developers of Aloe products will need to keep in mind. And it really comes into play if you have to give your allogeneic therapy more than once. And that may be the case because eventually allogeneic therapies are probably going to be rejected even in patients with compromised immune system. But hopefully they'll clear the tumor before they're rejected.
So I know we're running out of time, but I did want to get to the manufacturing side. I guess it follows on from this discussion. And I appreciate that every one of you is in different stages, but we'd like to just get a sense, even from a business perspective, how you're thinking about building capacity, partnering. Are you building it in-house? Even for some examples where you do have such a small amount of the cells available, and then you have to expand. How are you thinking about making that a scalable process?
Yeah, no, I can start again because my answer is very easy. We are tapping into the existing infrastructure. The answer is easy, but the practicalities are actually quite hard because due to the boom in the CAR T space, a lot of the established manufacturers have quite a lot of backlog, have waiting lists. And if you're kind of the new kid on the block, you might be not able to jump queues as some of the other people are. So it, for us, is a purely kind of business development operational issue to make sure that within a reasonable time frame, we can get into the clinic and kind of get our validation.
So I have to confess that I'm a little envious of Chris's 100,000 square foot. It's quite a large manufacturing facility. We don't have an in-house manufacturing facility, but what we did do is we designed from the get-go to have a fully closed GMP compliant manufacturing process. For those of you who've been in this field for a while, you may realize that a lot of people start out with research processes, they take them into the clinic, and then at some point in time prior to licensure, they have to adapt the process to a CGMP compliance. That's not trivial. And so we actually chose to take that pain up front develop a CGMP compliant process. But to do that, we actually partnered. We have some bespoke materials in our process. We have an antibody that's proprietary to us. We have a vector that's proprietary to us. Obviously, drug substance, drug product. We actually went out around the world and found different CMOs that have capability for antibody production, virus production, and cellular drug substance, drug product, and we're partnering with them. We started that process at the end of last year to enable an IND later this year to enable first-in-human studies early next year. So it's not, it's not a short process, but one goes through it, and it was the most efficient way for us to do it and ensure that we had GMP compliance right out of the gate.
Well, there's a paradox here because as a cell therapy developer, you know it's going to take a long time to scale up and to be at full commercial capacity if and when you hit the market. On the other hand, you don't usually have the resources to make that kind of investment that you really do need to make. So how do you manage that? And I wish I had the magic solution to provide you with. In our case, we got lucky because we found a couple good partners. So our strategy is with the Baylor College of Medicine, who are leaders in academic cellular therapy. They are manufacturing our phase 1 product for us. It is a GMP product. But there are some open steps, and there will be a need to scale it up. So while they're making that product for phase 1 and running phase 1, we're scaling up with the help of Cell and Gene Therapy Catapult in the UK. And so we'll have at least over a year to do that scaling up and development. So we'll be ready for larger scale clinical trials when they come. And you may have heard about that. I think Cell and Gene Therapy Catapult maybe is here. And that facility is in Stevenage, UK. And there's several companies in that facility, UK-based companies. Chris?
Yeah, as I mentioned, we've built our own manufacturing facility. I think it was certainly not without nervousness. At the time, 2 years ago when we did this, it was a substantial proportion of our available capital. But knowing how much we wanted to do and how broad our pipeline was, we felt it very important to be able to take our destiny into our own hands like that and actually put in that facility all the features that would allow us to meet the compliance rules that are needed for providing therapy to, for example, Europe as well as other jurisdictions all around the world.
Maybe you can share some with the panelists of your capacity. So I just wanted, we have a couple minutes left, so just wanted to see if anyone in the audience had any questions before we wrap up here. Okay, I'll ask one more. I think one of the other questions or conversations we hear a lot is just other strategies for cell-directed therapies, so thinking of BiTEs, for example. So maybe you can talk a little bit about that in terms of a potential competitive product or category, rather.
Yeah, I think the field of BiTEs has made quite a lot of progress, and if you look at some of the most recent data, some of the even first-generation BiTEs have shown comparable clinical efficacy and safety to some of the cell therapy products. And the question that we ask ourselves without knowing the answer is, like, how much better does a cell therapy product have to be in order to justify the very significant larger investment into the whole manufacturing CMC distribution aspects? And I leave it at that question because I don't actually know the answer.
I think BiTEs solve one problem. They solve the problem of targeting. But as we talked about earlier in this discussion, there are a lot of other challenges that face therapies that want to be effective in a range of malignancies, including solid tumors. So BiTEs bispecific T-cell engaging antibodies certainly have their place, and they're not without efficacy. But they do still rely on that potentially dysfunctional immune system to be effective, and they really only at this point in time, most of them only solve one problem, which is targeting. I think there are other challenges that need to be solved, and that's really where the benefit of a cell therapy, gene-modified cell therapy, comes into play over and above the capability of the BiTE.
All right, so we are officially out of time. Thank you very much. Also, there's a survey on your chairs. If you want to take some time to fill it out, we'd be happy to share the results with you. You can put them on the BTIG table out there if you don't want to fill them out now. Take them with you and do it later. Thank you very much, everyone. Thank you to the panelists.